So it is my pleasure to introduce Dr. Lorch. Dr. Lorch is a medical oncologist and professor of medicine and medical director of the Head and Neck Thyroid Programs, Northwestern Medical Group in Chicago. He became the inaugural director of the Thyroid Center is that Dana or Dana? Dana-Farber Cancer Institute in Boston. And he left Dana-Farber in 2021 to join the faculty of Northwestern University. Dr. Lorch is a ThyCa medical advisor. So I'm going to turn it on over to Dr. Lorch.
So welcome everybody. It's a pleasure to welcome you to the highlight of this conference. Anyway, so some conflict of interest. So when we think of, so the title of this talk is Immunotherapy in Follicular Thyroid Cancer. And I think if I just talked about follicular thyroid cancer and immunotherapy, it would be fairly brief. So I want to expand a little bit about immunotherapy and differentiated and not so differentiated thyroid cancer. So when we think of immunotherapy, then there are essentially right now, at least, there are two types of treatments out there. One are the checkpoint inhibitors, and the other are the T-cell or cellular therapies. And there are some advances in both. So the checkpoint inhibitors started around 2010, 2011, and they're very different from the traditional chemotherapy. They use your patient's own immune system to fight the cancer. So these drugs are not active directly against the cancer. Those only work in conjunction with a functioning immune system. And when you pick a disease for immunotherapy, you need something for these immune cells to latch onto, which means that the more mutations and the more abnormally expressed proteins or just substances are there on the cell surface and around those cells, the better, in general, immunotherapy works. So thyroid cancer, which is notoriously poor in terms of mutational events, is really not the obvious choice. So here you see about 60% or so are BRAF mutated, then you have a number of RAS mutated cases, and then a little hodgepodge of everything. But really, the main message here is that this is just a very bland tumor genetically. And if you compare this with other types of tumors, such as lung cancer, breast cancer, any smoking-related cancer, then you'll see thyroid cancer is actually, it's really hard to read, but that's over here. So there's really not much going on. So it's really, thyroid cancer is really not the obvious choice for an approach like this. Now, when you look at the more high-grade thyroid cancers, such as protodifferentiated or anaplastic thyroid cancers, and the picture changes a little bit. You do have more mutations that are known to convey aggressive biology, such as TERT, PIK3CA, and then also down there, some of the epigenetic pathways that are also activated. So intuitively, you would say, well, it's probably the more high-grade lesions that should respond to an approach such as immunotherapy. And if you then look at Hürthle cell cancers, now that's a type of follicular thyroid cancer. And if you look at the mutational profile there, then it actually gets really, really blunt, really scarce. There's really not much in terms of traditional mutations. Sometimes you see a RAS mutation. There's also a few cases here and there that may be BRAF mutant, but for the most part, there's really nothing on genomic sequencing. What has been demonstrated recently is that fairly recently, is that there are recurring mitochondrial mutations that seem to convey perhaps some contribute to the biology, and then there's also this odd near complete haploidization of the genome. So it's just to summarize, so the mutational landscape is generally fairly bland. More aggressive histologies have a higher total mutational burden, and prior chemotherapy may add to the mutational burden as well. So now with this, and the fact that there aren't really that many mutations, it's really not surprising that throwing a PD-1 inhibitor, which is pembrolizumab, just at unselected iodine refractory thyroid cancer is really not very effective. There were 2 out of 22 responses, and so for a total of 9%. So this is not very good. Now, if you look at the more high-grade cancers, such as anaplastic thyroid cancer, then such as here in this study with a PD-1 inhibitor called spartalizumab, it's not a drug that's clinically being developed, but it's very similar to pembrolizumab or nivolumab or some of the others. There was at least a 19% response rate, so it's already pretty much better. And if you look at the waterfall plot, which means that this is the radiographic response, then you see that there are lots of cases that meet and exceed the 30% response bar, which means like 30% shrinkage of tumors. Now, some further development of just PD-1, like combining it with a tyrosine kinase inhibitor, done in Germany, and originally and now confirmed in several trials that combine a multi-tyrosine kinase inhibitor with an immunotherapy drug. And there's a fairly good response rate, but overall still relatively small numbers. So we'll have to see whether this pans out in the long term or not. This is a study that combined a number of different tyrosine kinase inhibitors with immunotherapy, depending on their genetic profile. This was Maria Cabanillas' study at MD Anderson, done in anaplastic thyroid cancer, patients that were BRAF positive, RAS positive. Now, this would be the follicular component here, or these are typically then anaplastic thyroid cancers that arise from a follicular thyroid cancer, and then and then others, and those were treated with a PD-L1 inhibitor plus an array of tyrosine kinase inhibitors. And the first group, the BRAF mutated cases, did fairly well. There wasn't such a strong signal with RAS or NF1 mutated cases down here. And even though they did survive for a long time, but the response rate, which was the primary endpoint, was really not very different from what you would expect, and also not very high. And the third group also didn't do terribly well. This is the data for the BRAF group, and compared to historical controls, that seemed to be a bit better than otherwise. But this leads, though, to an approach that we did when I was at Dana-Farber. This is essentially what's called a microfluidic chamber system. It's a model system where you can test checkpoint inhibitors in a model, in a relatively simple model system. So you can use cancer cells from a mouse or from a human. You implant those in a 1 centimeter collagen cylinder, let them grow for a few hours. And if they grow, then you see these colonies here. And you can treat them then with checkpoint inhibitors. The nice thing is that in this microfluidic chamber system, the tumor microenvironment is preserved, and so you have all the immune components there to then also to see an actual response to immunotherapy drugs. This down here is a time lapse movie that work, I guess. Let's see if I can get this to work. Oh, no, it's not. No, it's not working. But if it worked, you would see that you have like lymphocytes, like immune cells, dancing around this focus of cancer cells, and this colony is actually then getting smaller. You can then measure cell death. You can also measure the release of cytokines, which is a measure of the intensity of the immune reaction. And that was, we saw that there was a bit of response to, or there was decent response to PD-1 inhibition, like the traditional nivolumab, pembrolizumab, spartalizumab. but that CTLA-4 was actually perhaps even more active in this disease. And that was the basis then for a study that we did that included, the main cohort were patients with differentiated thyroid cancer. That's included Hürthle cell cancers, follicular thyroid cancers, but also many papillary thyroid cancers and some poorly differentiated thyroid cancers. There was a group of medullary thyroid cancer and anaplastic thyroid cancer. Also, patients then received nivolumab or ipilimumab initially, like a PD-1 and then a CTLA-4 inhibitor. And then the other drug was added after 2 weeks, and the purpose was to tease out any differential effects of these drugs on the tumor. And for anaplastic, we started both drugs at the same time because this is such an aggressive disease, and we didn't want to lose time. And the results were interesting. So what we had hoped was to see a higher response rate than those 9% that were seen with pembrolizumab. And we did not see that. It was actually exactly 9% again, right here. These were the patients with differentiated thyroid cancer, the main cohort. Among patients with anaplastic thyroid cancer, though, we had 4 responders or 3 responders, but then 2 with stable disease. If you look at the waterfall plot again, there were the anaplastics are those blue bars over here. So there were quite a few responders. So that was quite encouraging. But now with when you look at the different histologies, such as Hürthle cell carcinoma, for example, which is again, which is a type of follicular thyroid cancer, or some of the mixed histologies, then you see that Hürthle cell cancers also seem to respond fairly well. We had 2 responders out of 7 cases, so that was also not something that had been observed before. And here are a couple of examples. So this is one case with metastatic anaplastic thyroid cancer and a nice response. And the patient, I think, is still in remission. And oops, let's not talk about that. Based on these results, we started out another study, which is with a bispecific monoclonal antibody. So this is essentially an antibody molecule that combines anti-PD-1 and anti-CTLA-4 activity in one molecule. So it's instead of 2 drugs, it's 1. And until earlier this week, we were enrolling quite nicely on that. Unfortunately, we just got shut down this week. So this protocol will go offline again because of some safety concerns that the company thought they saw, not just in our trial, but with other trials also with this particular compound. So this trial, unfortunately, is no longer an option right now. A Spanish group or a European group used a similar anti-PD-1, anti-CTLA-4 combination, presented that at the ESMO Cancer Convention a year ago. And again, a very similar design overall and same idea, and also saw pretty much superimposable results. So there was a response rate in differentiated thyroid cancer, wasn't great, but in anaplastic thyroid cancer, a response rate of 33%, which is actually exactly what we had also observed. And then other measures of success were also quite positive. So with this, I think it's fair to say that in an unselected population of differentiated thyroid cancer, the response rate is fairly low. In anaplastic thyroid cancer, the response rates are higher. Who knows? Perhaps the combinations of tyrosine kinase inhibition plus checkpoint inhibitors, that could be something that needs to be further explored. But beyond that, there isn't much with the checkpoint inhibitors going on right now. Now, briefly, oops, I thought there were a few slides about CAR T cell study. I guess it's an older version, but it's okay. So there's one study, or there's several studies out there now using CAR T cell treatment in thyroid cancer. There is the CAR T cells technology was developed also about 10 years ago, and it combines essentially an antibody that's implanted on the surface of a lymphocyte from a patient with the disease. And then patients receive some chemotherapy to suppress our own immune system and then get this engineered T cell reinfused. And the hope is that these re-engineered T cells will then attack the cancer. So this trial is open at MD Anderson, at Northwestern, and at City of Hope currently. But it's too early to say anything about activity. With that, I was hoping that we'd have a few questions.
Can you explain the manufacturing process you do at Northwestern, or do they fly it in from somewhere else? And how does that work?
For the CAR T cell study?
Yes.
Yeah, so this is a protocol that originated originally at Cornell. So the production facility right now is at Cornell. It's administered through a company called Immune. They will they run the trial. They have the patent on this technology. And the way this happens is that a patient gets screened. Then if they're accepted into the trial, they get this leukapheresis, so they get their white blood cells, their lymphocytes removed. They get sent to Cornell, where the T cell receptors are then re-engineered, and then they get their reinfusion about 4 weeks or so after that. It does require a 2-week hospital stay. The potential toxicities are cytokine storm or cytokine release syndrome, which can present very similar to sepsis. It can be fairly intense, and then neuropathy, which is also very, the mechanism is thought to be the same. There's just some, vascular damage that happens as a result of the intense inflammation, and patients have to be followed for a long time. But it's a one-time treatment, and that's what makes it attractive.
So my question is specifically for papillary thyroid cancer and BRAF mutated PTC. I mean, we know that the BRAF agents have been studied and they don't show as good responses as lenvatinib. But what are your thoughts on looking at PD-L1 or immune expression after somebody is tried on BRAF MEK inhibitor for BRAF DTC, and then add on immunotherapy at that time? Like, instead of switching to lenvatinib or cabozantinib as second line, what if somebody starts with DT alone and then adds on immunotherapy? What are your thoughts on that?
Yeah, I mean, it's it's something that's also being explored. I think it's not entirely, well, it's surprising in the sense that that treatment with like BRAF directed therapy is not more effective than it is. But then if you consider that we don't really know what the relevant targets are for multi-tyrosine kinase inhibitors such as lenvatinib or cabozantinib, so then I think there's just a higher chance of hitting something that's relevant. Now, the question is, could there potentially be synergy between a tyrosine kinase inhibitor and a checkpoint inhibitor? I mean, the data in anaplastic thyroid cancer suggests that there may be. Now, of course, there's also been a study where lenvatinib was combined with a checkpoint inhibitor, and for all practical purposes, the outcomes were exactly the same. So it's not obvious that there's going to be some potentiating effect at all. It may be just additive. Could there be a better response then at that point when they become resistant? Theoretically, that could be the case. Yes, so I mean, you know, how these decisions get made is a little bit of a mystery, and there's some leadership change at the company, and you know, and so on, and yeah, it's a bummer. And I was fighting to keep it open, but it's not going to happen.
There's a couple questions in the Q&A. So the first question is coming from Michelle. Michelle says, what would you suggest for immune therapy for possible 70%, I think this is Herthel, am I pronouncing that
Yeah.
right? Herthel cell thyroid cancer. The doctor says to remove the thyroid. Is there an alternative?
Yeah, that's a good question. So how big is it? Sorry, can you?
70%?
Yeah.
Herthel cell thyroid cancer. I'm not sure how big it is, but Michelle, if you'd like to, you can come on. Give me 1 second here. You can unmute yourself and talk to us. 3 centimeters, she says, but you can also unmute yourself, Michelle, if you'd like
Yeah.
to talk to us.
But-
Yeah, it's 3 centimeters.
Yeah, and the 70% number was, what was that?
That's they tell me it's 70% likely that I have Herthel cell thyroid cancer, but they told me that 2 years ago, and I'm still here, and
It's not, it hasn't grown very much. Yeah.
Yeah.
So, you know, Herthel cell is a little bit of an odd one because, you know, there are Herthel cell lesions and there's Herthel cell cancer. And, you know, so if you have a nodule and you stick a needle in and you get some Herthel cells out, it doesn't necessarily mean that this is Herthel cell cancer. In fact, the odds are that it's not, that it's a Herthel cell neoplasm. Now, ultimately, the way to decide what it actually is through surgery and then looking at it under the microscope to see whether there's actual invasion and so on and so forth. So
well, they did a thyroset gene test. That's why they say it's Herthel cell thyroid cancer, because they did that gene test.
Yeah. I mean, you know, a few years ago, there would have been a resounding recommendation for immediate surgery. Now, people have gotten a little bit more relaxed about these nodules, especially when they're very small and when they're not growing, to just watch them. And it turns out, or, you know, from the data that we have, it seems that that does not affect, you know, survival or any one of the hard endpoints. Now, you have to keep in mind, this has been done mostly in papillary thyroid cancers, which is really a little bit of a different animal. You know, Herthel cell cancer, if it is cancer, Herthel cell cancer typically does not respond to radioactive iodine. And, you know, and it's, even though it's relatively rare, it's only a small percentage of all thyroid cancers, but it's about 30% or so of all iodine refractory thyroid cancers that occur. So it's with Herthel cell, it's a little bit of a tougher decision than with some of the other cancers. I mean, I think there's probably, especially if this is not, you know, especially if this is not growing, I think it's interesting to just watch this. But I don't think you can't rule out that there might be something that, you know, happening in the meantime, and these things can metastasize, and when they do metastasize, they tend to then cause problems down the road.
Do you mean when you say it's not growing, you mean the nodule's not getting bigger? Is that what you mean?
Correct. Yeah. If it stays the same by size, but you never know what's going on inside of it. So, you know, I think it can be done. It's justifiable. But you know, classically with a 3 centimeter lesion, you would generally take it out.
Yeah, they say it's as big as my thyroid, the nodule is.
Yeah. So I mean, you know, again, it's possible to just watch it, but there is, I don't think anybody knows for Herthel cell specifically, but you know, I would say that there is a risk. If you're, you know, willing to take that risk, I think it's fine.
Okay, thank you very much. Thank you. I appreciate your help.
Yeah, sure.
Thank you, Michelle.
Thank you.
Another question. Are there any ongoing personalized studies with monoclonal ABs or someone working on it? What is the update for something like this? Something like Ralph Steinman did.
Ralph Steinman. I don't know who he is. But so, I mean, the class of monoclonal antibodies is very large, and all the immune checkpoint inhibitors are, or most of them fall into that class. They're usually infusions of these monoclonal antibodies. There are, there was our trial that was testing this, that we were testing in thyroid cancer with this bispecific PD-1 CTLA-4 antibody. There are trials ongoing currently combining a tyrosine kinase inhibitor such as cabozantinib plus a PD-1 inhibitor or PD-L1 inhibitor actually. There's a trial with lenvatinib plus tyrosine plus checkpoint inhibitor. So there are several, but right now, to my knowledge at least, there are no phase 2 studies in thyroid cancer that are currently conducted with other monoclonal antibodies or monoclonal antibodies that are other than checkpoint inhibitors.
Ralph Marvin Steinman was a Canadian physician and medical researcher at Rockefeller University who in 1973 discovered and named dendritic cells while working as a postdoctoral fellow, won a Nobel Nobel Prize in physiology, and also had pancreatic cancer.
Okay. Yeah. So dendritic cells play an important role in the immune response, and that includes response to immune checkpoint inhibitors or other cell therapy based approaches. The purpose of these CAR T cells is to then, once they find their appropriate target, to then also interact with dendritic cells and get them to release some cytokines and so on and so forth. So dendritic cells are really at the center of the immune system, or it's one of the main components of the immune system. So they do play an important role. There is also there are efforts to use dendritic cells directly as cell therapy. To my knowledge, it's not done specifically in thyroid cancer, but there are several studies using this or dendritic cells, natural killer cells, and some of the other components of the immune system, try to harness those to treat cancer. I don't think specifically for thyroid cancer, but yet at least. But yeah, so it's that is a very well-known part of the immune system and does play a big role. Yeah, so they started actually in solid tumors as far as like lymphomas goes, and like things like CLL, so chronic lymphocytic leukemia, which is a B-cell lymphoma / leukemia, and then it also started in some pediatric leukemias. These were the first 2 indications. That's they've become a standard of care treatment in those illnesses. So to bring these into the solid tumors has been a little bit of a challenge, and nobody understands fully why. But the situation between liquid tumors and solid tumors is just fundamentally different. You have in liquid tumors, you have these single cancer cells that are floating around. They're readily available. They can be targeted from multiple angles, like just sort of sterically speaking. Whereas in solid tumors, you have some extracellular matrix that these tumor cells might be embedded in. The tumor cells, the tumor centers are often hypoxic and are not very well perfused, which means that there's not much blood flow, which means that there's not much oxygen. So it's a much tougher environment for the immune system to actually do anything. So that in solid tumors, so far, there has not been a huge success across the different tumor types, but there are some exceptions. There are some cell therapy studies, for example, in HPV-related disease. So now that's not thyroid cancer, but so the concept seems to work in solid tumors also, but it's just requires more. It will require more research to figure that out completely. I mean, there's also starting 10 years ago with the first generation of CAR T cells, the technology has also progressed. So initially it was essentially just an antibody attached to a regular T cell receptor portion, intracellular portion. Now, several other as the generations of those molecules progress, or this treatment progress, and currently I think it's like 5th generation or so. There are now components that are that provide, for example, that actively produce cytokines in the T cells, for example, or that provide other mechanisms to enhance the immune reaction. So I mean, it's an area of very active investigation. I think anaplastic and poorly differentiated thyroid cancer is a pretty good target for this because it is a very immune sensitive disease, just like melanoma and certain lymphomas. Yeah, so the question is, what's the role of, if I understand this correctly, what's the role of glucose, the glucose metabolism in the growth of thyroid cancer cells? And is there a way to use this therapeutically by starving thyroid cancer cells? Yeah, so obviously there's a lot out there about glucose, and then leading to certain dietary restrictions. Don't eat a lot of sugar, or no dairy, and so on and so forth. So there's a lot of these ideas out there. I mean, thyroid cancer cells do like to eat sugar. We know that from PET scans. The more aggressive they get, the more they like sugar, and the more these lesions eventually light up on on a PET scan. Now, to starve these cancer cells is a little bit of a challenge, because generally, the glucose levels in our blood is pretty, those are pretty tightly controlled. So it's relatively hard to influence the glucose level, just by avoiding sugar, for example. I mean, our body generally needs some glucose. I mean, the brain almost exclusively runs on glucose, and so do some other things. So, I think it's ultimately, can you use this therapeutically is something that's unproven. I mean, in some ways, intuitively, it would make sense. Personally, my feeling is that it's probably a little bit, it's more complicated than just stopping to eat sugar. You could then starve the cancer, or somehow harm the cancer more than the rest of your body. So I think it's ultimately it's unknown. Personally, I'm a little skeptical whether that's a viable strategy. I mean, I think a regular, healthy, varied diet, I mean, is probably the best way to deal with that. I think it's difficult to influence these levels in a major way. No, it's not. No, I mean, there are some targeted therapies, not specifically in thyroid cancer, but in other cancer types, that are trying to interfere ultimately with glucose metabolism, and I'm sure that there is probably also an effect of these tyrosine kinase inhibitors on glucose delivery. If you're restricting blood flow into these tumors, I'm sure it has some impact on the delivery of glucose, and it has to. But is that one of the main mechanisms? Probably not. And can this be used therapeutically? Well, maybe, but I don't think we have the tools yet. And I think simply avoiding eating sugar is probably not the answer. Yeah, no, I know. I mean, there are some data, there are actually some data, even animal data in thyroid cancer that a ketone diet, at least in mice, seems to slow down the growth of anaplastic thyroid cancer cells. I mean, these are all fairly artificial systems. Now, how much can you translate that to or what does that tell you about what's happening in an organism such as us, who that lives 80 years, weighs too much, but compared to a mouse and so on. I mean, it's just very different, and I mean, I think there's probably a connection, but we don't have. It seems to me we don't have the tools yet to harness that. Yeah, yeah. So there are several of these trials that also had medullary thyroid cancer cohorts. I mean, one was at MD Anderson, right? There was a trial, and my understanding was it wasn't overwhelmingly positive. In our cohort, certainly we didn't see much of a response either with nivolumab and ipilimumab. The Spanish study that I mentioned, they did see, again, among a handful of patients, they did see, I think, 2 responders. So their conclusion was that it may be active. In our trial, I do recall one patient who was quite young and had very aggressive thyroid cancer, medullary thyroid cancer. And we saw a drop in calcitonin levels for a little bit, and it slowed down for a little while. Now, again, his cancer was unusually aggressive. So I think, again, if you picked the worst medullary thyroid cancer cases that you can find, you might end up with a higher response rate ultimately. But it's certainly not overwhelmingly positive. And again, these are typically tumors that are driven by just a handful of, or maybe just 1 or 2, driver mutations. So it's not really a disease where you would that would be the obvious choice for an approach like that. There's a CAR T cell study that's going on at the University of Pennsylvania. That's also using not a red-related, actually totally unrelated epitope as their target. And again, that works beautifully in mouse models. It's clinically, at least so far, hasn't been successful yet. But again, as time goes on and these treatments, CAR T cells get better, there might be an application at some point. But so far, I think, with the exception of that one European study, I think the experience has been somewhat disappointing. Yeah, it does. I mean, the spartalizumab study found that among patients with a high PD-L1 expression, now PD-L1 expression is the expression of the PD-1 ligand, and that's known to be a fairly decent predictor of response to immunotherapy treatments, to checkpoint inhibitors in particular. And although the correlation varies a little bit, there's not a direct correlation. It just makes you it's a little bit more likely to have a response if the tumor has a lot of PD-L1 expression. Now, so for example, in the spartalizumab trial, which was in anaplastic thyroid cancer with a 19% response rate, among patients who had a higher positive score, like a higher PD-L1 expression, there was then a 30% or so response rate if you selected for those. And we saw something similar in our cohort. So I think, yeah, there is a correlation, but it's not as strong as you would expect. Yeah, we had a few, and there was also a connection between RAS mutations and response, but again, it wasn't something that would like immediately suggest that having a particular RAS mutation that you then automatically have a response. In the spartalizumab, yeah. Yeah, I mean, it should, again, I mean, the question ultimately is, how, should we test for PD-L1 expression at all? Or should you exclude somebody from treatment with immunotherapy if their PD-L1 expression is very low? And in my opinion, the answer to that is no, I don't think we should exclude people. There's still the possibility for them to respond. It's just somewhat, it's comforting to know that there's going into this treatment, and also considering that sometimes these treatments can be quite toxic. It's good to know that if a particular patient has a good chance of a slightly better chance of responding, but I don't think it would be as a predictive marker. I think it's the correlation is just simply not strong enough. In that same paper, what they also found was that the signature of T cell activation also correlated, and perhaps correlated more strongly with the response versus versus those who did not respond or did not have that signature. I think that makes a lot of sense. It's sort of one step closer to actually, getting an idea about the microenvironment and what's actually happening between these cells. I mean, these interactions are just so complicated and involve so many cell types and cytokines that it's difficult to predict that just based on a PD-L1 score alone. I mean, that depends, but I mean, it sounds like this patient probably doesn't have a lot of options. So yes, I would consider nivolumab plus ipilimumab, because there you have the possibility, at least theoretically, exists that by activating T cells through CTLA-4 blockade, that you have something that goes beyond PD-L1 or the PD-L1 mechanism. But I mean, would I not give it to someone? No, I wouldn't. I would definitely go ahead and do it. The problem is with frequently is coverage, and increasingly so, I find. Yeah, I mean, that's also what I frequently do. So if if they have stability on lenvatinib plus pembro, for example, then I'll obviously I'll just continue. Once they progress, I may just add everolimus. So for RAS mutated cases, I often add everolimus, and I've done the quadruple combination of everolimus plus lenvatinib plus nivo-ipi, and I mean, it doesn't add much in terms of toxicity. And I've had people who then had at least stability for a little while longer. I mean, no like dramatic response, but I did see some, there seems to be some activity. Now, would that be different from just nivo-ipi plus lenvatinib or lenvatinib alone? That's sort of unanswered questions, and this is just anecdotal stuff. But usually, many times you can find something to keep them stable for a little bit longer. But ultimately, if there isn't this great overwhelming response to checkpoint inhibitors right away, it usually then becomes a long battle, which you ultimately lose. I've done it off protocol. It's probably, I don't know, it's probably bad practice, but I've done it. I mean, in a disease like anaplastic, exactly. I mean, it's not. There's no such thing as bad practice, I guess. And it's pretty well tolerated overall. All right. Thank you very much.
Thank you, everyone.